Sorry for the, I don't know what happened in between there. Okay, so as Olga says, I'm going to give you a little bit of an introduction to data independent acquisition. And then after this, Lindsay is going to give up sort of a follow-up, a practical tutorial on how to start to interpret this type of data. So I'm excited for that. You should definitely see both of these sessions side by side. So as Olga said, I'm just starting today my appointment at Ohio State University. So I have an email address down here, but I also have my personal email just in case if you need it. Also, the slides for the this session are actually on the website, but these are Creative Commons licensed. So if you want access to the original PowerPoints, feel free to email me then, and I'll try to get them to you in a reasonable way so that you can look at how the slides were working and then dig into them a little bit more deeply or potentially present that material on your own afterwards. So just to kind of start off, I like to give a slide about speaker disclosures just to explain to you where I come from. So as Olga said, I'm now an assistant professor at Ohio State University, but I also have a founder's position at Proteome Software, and I am a shareholder there. Most of my research funding comes from the NIH and from a little fund associated with IARPA. So with that in mind, I kind of wanted to start us off easy. So just to explain to you a little problem that I had been working on recently, I've been interested in studying how infection works in cells. And so I've been analyzing proteins from viruses in human cells. And so how would I do a problem like this, right? The first thing that I would do is I would take those proteins and I would linearize them. I would stretch them out and denature them. And then I would digest them with a digestion enzyme, in this particular case called trypsin, and cut them up into little pieces that make it easier to analyze with a mass spectrometer. And then finally, I would try to analyze that list of peptides that are generated. You know, most of those peptides are going to come from, in this particular case, the human cells, and the peptides that I'm interested in are essentially like needles in a haystack. They're hard to find. So in a problem like this, which is in a sense, very similar to virtually every type of proteomic problem, rarely are we interested in the most abundant albumins of the world. We're really interested in the stuff that's difficult to see generally. So in these particular cases, we have a bunch of tools to be able to do that. So I want to lead you through some of the ways in which we analyze this type of data. And the most common way in which we analyze this type of data is called data-dependent acquisition. So here in this framework, we're analyzing peptides as they elute off of an HPLC, separated in time over retention time, and separated en masse by the mass spectrometer in precursor space. So this would be a circumstance where each one of these peaks represents a different type of peptide that is in my sample at any given time point. So the way DDA works is that over time, we see a peak come up, we select it for fragmentation, we collect a fragment spectrum, and then we stick it on an exclusion list where we ignore it for a while. This helps us be able to dig deeper and deeper into the proteome. And so then we'll go to the next peak, and then the next peak, and then the next peak, and the next peak, and the next peak, and we'll sequence all of these guys and give us an idea as to what's going on at each of those different peptides. So how does this work in the mass spectrometer? We collect these precursor scans like we showed you before and pick peaks out of this. In this particular case, I'm showing you the top 12 peaks that the instrument would pick based off of intensity. And then all of the rest kind of get left out. And then again, all of these 12 will get stuck in an exclusion list so we can sample them over time, sample other peptides over the same time period. But there are some complications with this approach. So right here, I'm just showing you kind of a screen capture of what 60 seconds of peptides looks like. So this is, if you're looking at a precursor scan over time, you can see the peptides rise and fall over time. And so you can watch them with their relative intensities go up and down. And there are some regions of this proteome where you see these peaks that rise up and down, that they look like normal peptides, but they're just never going to get to the threshold at which we would ever try to send them for a sequencing scan. So there are lots of these types of peptides that are really valuable things that we'd be interested in detecting that we just won't ever be able to see. So if I take this, again, this 60 seconds of peptide screenshot and sort of show you all of the peptides that, in this particular case, this is on a Q exactive. These are all peptides that are above, that I've colored here, are peptides that are above 1E7 in intensity. So that's a very high threshold in terms of its peak area. That's much higher than we usually set for our normal dynamic exclusion thresholds, which tend to be around E3 in terms of their, so 4 orders of magnitude separation between them. So there are a lot of peptides. These are all of the peptides that are in that range, but already there are more peptides in this time point of just 60 seconds than we have scans, than we have MS2s that we can allocate towards measuring these types of peptides, already within E7 in this particular case. So we're saturating in terms of collecting MS2s very quickly. Most of the time we saturate almost instantaneously. And so let me show you what that means. So up at the top here, I've got a normal sort of tick trace where I've got retention time over time and then precursor tick on the y-axis. This is just showing you where the density of peptides are eluting off of my mass spectrometer. And you see quite quickly on the bottom graph where I'm looking at the number of MSMS collected in purple in a normal DDA scan, we hit the maximum point, the maximum number of MSMS measurements almost immediately, very early into the gradient. And after a certain point, we still are collecting huge numbers of MSMS even out to the very end of the gradient. Now, if we look at the number of peptides detected, so the number of PSMs measured in this time period, what does TIC stand for, Jennifer? Great question. TIC stands for the total ion current, which is basically a measurement of the amount of ion signal that we're seeing in the mass spectrometer at that time point. So if that signal goes up, then we have more and more peptides at that time point. It's, instead of measuring a single peptide, we're actually summing all of the signal from all the peptides. Does that kind of make sense? And PSM, what does PSM stand for? Peptide spectrum match. So these are the number of detected peptides that we're actually seeing at any given time point. So in this particular case, at time point 30, we're detecting about we're collecting a full number of MS spectra, but we are only detecting about half of those spectra as actual peptides, or assigning to actual peptides. So one of the things that you can see that's really interesting here is that as you get into a sparser part of the gradient, that's where you're seeing fewer and fewer actual peptide signals from the tick trace. We actually identify a higher percentage of peptides in our total number of peptides as those PSM. So the green bars are higher than in that early section where you've got lots and lots of peptides based off of the amount of tick signal there. And so what's going on here? Actually, what we're seeing here is a circumstance where the measurements we're making aren't actually of individual peptides. And we are actually making collecting MSMS spectra that contain multiple peptides at the same time. And because of that, it makes it very difficult to make detections from those peptides. And this is just because when you have more and more peptides, the sort of space that you're able to sample gets more and more congested. And so when you select out a particular mass for MSMS, oftentimes there can be other peptides at those same points. So how do we sort out impurities by seeing these graphs? This is an excellent point. So all I'm showing here are +2 and +3 signal peptides. So there are impurities that are +1s that are usually sugars or other types of small molecules that could definitely be a potential problem here, but that's not actually what's going on here. The instrument automatically filters out any peptide that doesn't have a +2 charge or higher. So we know that these are measurements that we should be able to detect peptides from, but they're just too complicated to be able to make those measurements because they're coming from multiple peptides at the same time. So DDA, I think, is a really excellent tool for doing this type of measurement. And yes, you could increase retention time for doing this type of measurement. That's a good question, Christopher. But that said, the more you increase retention time, the more you have to spend time on that specific sample and not on other samples at the same time. So you can stretch it out further and further, but that still also doesn't make it such that if two peptides have somewhat similar chemical properties, they will still fall on top of each other, even if there is space between those peptides and other peptides. So you can make the problem a little simpler, but it still has a asymptotic potential. So just sort of thinking about DDA, we are making these fragment ion measurements, and we're collecting MS2s about them. And yes, we can do some little tricks about decreasing isolation window and all that kind of stuff, but these still realistically still let in other types of peptides at the same time. So unless you're using an orthogonal type of separation, say, for example, ion mobility, you're almost always going to get these very convoluted spectra where you see multiple fragment ions from other types of peptides at the same time. One way to think of this is anytime you see noise in your MS2, that noise actually comes from other peptides in the background. So there's definitely a lot of interesting signal that exists that we're not able to capitalize on because of the way we're selecting precursors and trying to assign a single peptide to each spectrum that we're measuring. So another strategy for trying to do this is trying to analyze samples with a technique called PRM. And if we're interested in monitoring these low abundant peptides, so for example, in this particular case, you can see that the dark blue and the light pink peptides were just never sequenced for MS2s. We can actually target specific masses that go after individual peptides that we're interested in. And rather trying to collect a single MS2 to represent that peptide, we can actually collect MS2s over time, one after another, sequentially over time. And this kind of gives us a way of measuring the peptide at its apex, and by being able to see a peak shape associated with it, we can be very confident that that peptide is actually present. We also guarantee that we're going to select the peptide of interest. So for example, in this particular case, that blue peptide. So this is a way for me to select out, for example, all of my virus infection peptides to be able to measure those very well. But one of the things that is a potential problem is I'm interested in measuring all of the viral peptides, all of the sources of viral peptides, not just selected ones that I'm going after. And so, yeah, Julia, you make a very good point. We do have a limit on the number of masses that we can assign in a PRM experiment. Typically, in a normal PRM experiment, we're only really able to analyze something between 150 and maybe 300 different peptides at a given time point, corresponding to maybe 50 or 100 different proteins. So we're not able to measure an entire proteome here. We're only able to measure a handful of peptides belonging to a handful of proteins. So that's definitely a complication of PRM that makes it limited in some circumstances. So just to kind of reiterate, PRMs are really great for making deep measurements, but they are a little bit complicated in terms of trying to measure an entire genome. So if I'm interested in measuring an entire genome, we actually need to ask the question, is it possible? Are there mechanisms to target every single protein? And there are some kind of clever ways that you can run PRMs to be able to do that. But what I want to do is talk to you about other way of thinking about this, which is data independent acquisition. So just to kind of get to a content slide, here I'm looking at DIA, and I'm going to talk to you about how we're acquiring this type of data, and then what types of measurements we're trying to make, and specifically thinking about it from a PRM perspective. Because instead of trying to do a DDA-like acquisition, we're actually trying, with DIA, we're trying to do PRM-like acquisition, just in a global way. Then we're going to talk about how we're going to analyze DIA data with libraries, some of the complications around measuring DIA data in experiments, in certain types of experiments. And then we're going to talk a little bit about some of the best practices for setting up a DIA experiment in your lab. And then finally about how we're going to analyze data if we only can acquire DIA data, which is oftentimes the case, and how I frequently am acquiring data these days. Okay. So just starting off at the beginning, what is DIA? And it's basically trying to take a PRM and expand it out to a global acquisition strategy. So again, thinking about that PRM type of measurement we've got here, if we're try to section that off as if we're looking at it from a DIA perspective, instead of selecting a specific peptide to measure, we're selecting a precursor mass window. So for example, here we would be selecting the mass window from 475 to 500 MZ and from 500 to 525 MZ. Okay. And so what's going on here is that we are actively co-fragmenting multiple peptides at the same time that come within the same wide window in this particular case. So the MS2s that we're collecting, the individual mass measurements, are extremely complicated. They come from oftentimes 20 to 100 to even a couple hundred different peptides at the same time in the same scan, in the same MS2. But one advantage here is that we get time dimension information. So we can monitor these fragment ions over time as they rise and fall. And so then the goal of a DIA experiment becomes to select out from those complicated interfering signals, which are the fragment ions that are associated with a specific peptide. And then all of a sudden we're looking at data that's much more similar to PRMs, and we can think of it from a targeted perspective, where we're making these measurements of normal fragment ions across time. In these particular cases, when we look at these things, we're thinking about peptides eluting over around 30 seconds or 60 seconds or therefore. So what are the kind of constraints that we're thinking about here? So there are some compromises that we're trying to deal with, and one of them is the fact that each of these peptides elutes over 25 seconds or so. And so the instrument can only scan so fast without sacrificing those types of measurements. First, we need to be able to collect good MS2s. And so good MS2s, it takes about 10 to 20 hertz on Orbitraps. You can get up to around 100 hertz on some TOFs, but as you start to speed up, you actually are dealing with fewer and fewer ions at any given point. So the actual MS2s that you're collecting are not quite as robust or not quite as accurate. So you're actually limited in terms of the scan speed at which you can acquire data. And usually we're thinking around 10 to 20 hertz, at best around 100 hertz. And the other aspect of this is you really need 6 to 8 measurements across a peak to be able to describe the shape of that peak. So you need to collect a lot of scans to represent every single peptide. And so there are a lot of compromises that we make relative to PRM. And one of them is that we have to accept much higher levels of interference by widening our precursor windows, as I said. So rather than targeting a specific mass, we're actually targeting a range. So in this case, 25 M over Z window. It lets in a lot of different peptides simultaneously. So we have a lot more interference in our measurements. The other aspect is that we have less time to sort of spend on each mass spectrum. So we can't just like sit and acquire on that mass spectrum for half half a second or something like that to be able to get the best signal. We actually have to run through these scans relatively quickly. So as I said, in the order of 10 hertz to 20 hertz to 100 hertz, in that kind of range. So the other aspect is that we kind of need to limit what we can measure. So rather than being able to measure any type of peptide, usually we have to think about a narrower window scheme where we're only measuring peptides from 400 to 1200 or 400 to 1000 or something like that. So what do these compromises actually look like? So what I'm showing you is measurements that I collected on a QE for PRM, where I get really good signal on the across the peak for these 5 transitions, you get really nice signal. And if I look at that same data with DIA, you know, I get I run up into some different kind of constraints. You know, one of them being that there's more interference and that there's a lot lower signal to noise, and also there's less points across the peak. But realistically, these types of measurements are almost identical. You can see that the maximum intensity of the PRM, again, goes up to 10 times 10 to the third. And again, you're seeing the same type of signal in the DIA experiment. It's really the same type of measurement. So in that regard, we can actually analyze this type of data, thinking about it as a targeted experiment, and we get a lot of benefit out of being able to reuse some of those concepts. So Lindsay later is going to show you how to analyze DIA data using Skyline, as well as some other potential tools that center around this sort of targeted perspective of trying to interpret this data. So what does DIA data acquisition actually look like? So as I said, it's comprehensive acquisition where we're acquiring a precursor spectrum, and in this case from, say, 400 to 1200, as was with the original Swath papers or DIA papers. And you collect a fragment ion scan versus a reasonably short range in that precursor scan, and then another, and then another, and then another, and eventually you scan through the entire window. You'll go down and collect another precursor scan and then another set of fragment ion scans. And the goal here is that you want to get around 10 points across the peak for a 25-second peak. So you need your window width. So that's the distance between each fragment ion scan and each other fragment ion scan to be around 2.5 seconds. So, so they need to happen in relatively fast order. So all of those, the precursor scan and all those fragment ion scans, have to happen in that same type of cycle time, around 3 seconds or 2.5 seconds. So let me take that data, that graph, and kind of compress it back. So now I'm just sort of shrinking it back so that you can see it a little bit easier. And if we zoom into one of these particular fragment ion scans from 400 to 75 to 500, we can look at this peptide here, LSGG, that we can see a really nice spectrum for. This would be as if we were thinking about it from a DDA perspective. And the reason, Hugo, why we're looking at 2.5 seconds is, again, we need to get 10 points across a peak that is 25 seconds wide. And so if you divide 25 seconds by 10, you get 2.5 seconds. Okay. So if you think about this, we're actually acquiring scans, multiple scans, one after another, to describe the shape of the peak. So rather than just looking at a single MS2 showing this peptide, we actually can see lots of MS2s describing this peptide. So we had tons of information in this sort of three-dimensional space where we're thinking about mass over charge and retention time, not just mass over charge and intensity. So mass over charge, retention time, and intensity. So again, if you look at this from the angle, you can actually extract out all of the fragment ions that are associated with the peptide that we're interested in and make a measurement like we would normally do with PRMs. And so this approach allows us to kind of ignore all of the peaks that are the bigger peaks, the gray peaks that are essentially interferences from other types of peptides. Okay, so there are some complications here, and we've already alluded to a bunch of those. But thinking about this again, within the framework of DIA, all of those mass measurements over and over, one important thing is that when we're making these measurements, the ions that we're measuring are actually not centered in that window necessarily. They can be anywhere in that window. So they can come from the very early part of that window, so closer to 475 or closer to 500, and they can be of any different charge. So when we're collecting an MS2, the ions that we're taking a measurement of aren't centered, and they're also not collision energy optimized for that specific mass over charge. So the fragmentation that we get is not exactly the same as if we were doing DDA, where we can sort of optimize what the appropriate collision energy is on the fly for any specific peptide, because we're isolating a specific peptide or trying to isolate a specific peptide in a DDA experiment. So that's issue number one. Issue number two, as we've already talked about, there are usually multiple peptides that we're trying to measure at the same time, co-eluting at the same time. So the fragment ions can overlap and sometimes can be exactly the same fragment ions, particularly in cases of low mass fragment ions. The third thing is that we have suppression at the edges of windows. So for example, at the beginning of that 475 and at the end of that 500 mass range, we actually have essentially a trapezoidal transmission efficiency. So not all peptides are actually put into the mass spectrometer in the same type of way. Or at the same intensity. So for example, that red peptide on the end might not get as much signal actually put into the mass spectrometer as that green peptide in the middle. Okay, so so let's look at a DIA data set. So what does this type of DIA data actually look like?
To interrupt, there is a question. Yep. Could you comment on the choice of the specific value for the ppm?
Oh, so in that particular case, when we were looking at a particular peptide, that was just the ppm of the mass accuracy for that peptide measurement. So that's really all it represents. It doesn't actually represent anything. It's just what was the ppm accuracy for that mass measurement.
Sorry, there was one more question, Brian, that I thought was really good. I think you're going to get to it later, but what do you think about isolation windows overlapping, having more peptide identification? So overlapping DIA windows, more identifications? Yes, no?
Yeah, so we're definitely going to talk about overlapping windows later on. So that's a fantastic question. We'll get there, definitely. And the last question, can I collect DIA data for discovery using the same data as for a targeted experiment? So what we are trying to do is actually do discovery style experiments just with essentially targeted data, right? So we're trying to treat this as a merger between those two different approaches, between a normal discovery acquisition, but specifically from a targeted framework. So the type of data that we're acquiring looks like targeted data rather than looks like DDA data. Okay, but it's still trying to be a discovery platform like DDA. Okay, so what does some of this DIA data look like, and how messy actually is it? So here I'm looking at a specific HHAA peptide from superoxide dismutase in this particular sample. And so if I show you all of the extracted fragment ions across time, each one of these peaks, each one of these colors represents a different fragment ion in that particular peptide. And I'm just extracting all those fragment ions out over time. So for example, if that first fragment ion or those first couple of fragment ions have a mass of say 475, here I'm extracting out 475 across the entire retention time to show you what's going on. So the first question is, where is this peptide? It's actually pretty hard to figure out normally, sort of naively, where this peptide is. And so let me just show you, it's right here. It's not the most abundant fragment ion associated with this peptide. It's actually one of the smaller fragment ions. But the way we know that this is the right peptide is we can look at the combination of multiple fragment ions together and the presence of multiple fragment ions. Even if you sum up these signals, they're going to be less than the most abundant signals. But they are, there are a lot of signals all coming out at the same time. So we can say, because all these fragment ions trace together, we can be pretty confident that this peptide is here. Now, this looks like a great example. Obviously, DIA works in this particular case. But let me show you this other peptide, this FNGG peptide. This is from, again, superoxide dismutase, exactly the same protein in exactly the same sample. So these should be equimolar in their abundance. So they're on the same column at exactly the same amount. And again, where is that peptide? It's actually in this grass here. But when I show you the region around here, it's kind of complicated. actually, what is the peptide that I'm actually looking at here? Is it the peptide on the left, or is it the peptide on the right, or is it the peptide in the middle? It's really difficult to say what is the right signal associated with a specific peptide. And so let me just show you, it's this particular signal, even though that's the one that looks the messiest. But how do I know that? And I know that because I can use information from previously acquired data. So I can use information in a library where I know the fragmentation of this particular peptide from a DDA experiment, a past DDA experiment. And I can look in my sample at that same time point and extract out all the fragment ions associated with that peptide. And lo and behold, it looks beautiful in terms of the integrations for all of those guys, where I've got high Y8s and Y9s, Y2s. I've got, again, a nice ladder from Y10, 11, and 12. And then all of the rest of the ions are lower than that. So I can be really confident that this is the right peptide based on those fragment ion intensities. But when I actually look at the underlying data, this is what the underlying data actually looks like. This is at the apex spectrum. This is the signal at the apex spectrum. This is the highest signal for this peptide. It's still hidden in the grass in this complicated spectra where I'm actually measuring, again, 20 to 100 different peptides at exactly the same time. So if you were trying to interpret this from a DDA perspective, you would end up kind of like making it would be very difficult to make this interpretation of this as being the specific peptide being present here. It's actually a much lower signal. Okay, so and we'll talk about, Raja, we'll talk about lots of different strategies for trying to think about analyzing this type of data. We'll get to that in a bit. But so one of the things that I can do here is looking at the fragment ions that are associated with this particular peptide. I can say that some of them are interfered with, say the ones in red. Those are just kind of not very useful in terms of making this particular measurement. The ones in yellow rise and fall with the peptide, but they also show some level of interference as well. The ones in green, we can say, those particular fragment ions, even though they're not the most intense fragment ions, because they rise and fall with the shape of the peptide, we can be really confident that that's what's actually going on here, that we actually are seeing this type of peptide. So Bowen, we're going to talk quite a bit about a library generation in a bit. So realistically, this DIA data is actually extremely hard to interpret when you're thinking about it from this way, this sort of naive way of trying to extract out the data. So we have a bunch of new approaches that we're going to get to for why we would actually, how we're going to extract this type of data out. But the first thing that I wanted to ask is, why even bother collecting this data if it's so hard to interpret? And there are some really interesting advantages. So when we're actually trying to analyze DIA data, we see lots of interesting features in the data that we don't see with DDA, because we end up making mistakes oftentimes with DDA. So one of those, for example, is getting an early DDA mass trigger. So for example, this particular precursor mass, we see, again, rise and fall as we would normally expect to see. We would trigger it from MS2, but there's actually no signal for this particular peptide. And we can see that there is another peptide that comes out just a hair later, less than half a second later. We can see that, or half a minute later, we can see that second peptide again come out with great MS2s associated with it. But we would never measure this with DDA because we would have stuck that peptide on a dynamic exclusion list. So another aspect of this is that with DDA, we find that the precursor signals oftentimes can be very complicated. They can have a lot of interference because it's only 3 measurements that we're able to make. We're able to make a monoisotopic measurement, a +1 and a +2 measurement. Again, the blue is the monoisotopic, the purple is the +1, the red is the +2. These are typical skyline colors that you'll be comfortable with later after Lindsay's presentation. But here we can see that there's these precursor signals. While they kind of line up with the peptide, they don't line up with each other. That indicates to me that all of these signals are heavily interfered with, and they actually come from multiple peptides at the same time. So if you try to integrate the precursor signal for this peptide, you'd end up with kind of a mess in terms of the actual measurement that you're making. And the third one is even the most interesting of all. We see a lot of peptides, actually, a reasonably significant number of peptides that don't have any precursor signal at all. At all. And so these are peptides that just go completely blind in the mass spectrometer, but we can see really great fragment ions associated with those particular peptides. And so what's kind of going on here? One way to think of this is if you're acquiring normal DDA type of experiments, you're taking all of the peptides that are coming off the column at exactly the same time, you're passing them through a quadrupole, and then in this particular case, I'm showing you with an Orbitrap, but the same kind of principle occurs in other types of instruments too. In an Orbitrap, you take those peptides and you put them into an HCD cell where you do some fragmentation, and you make the Orbitrap measurements of those particular peptides. You'll see a bunch of those. So in this particular case, we're actually just using the HCD cell to store the peptides. So we can accumulate, in this particular case, up to about 1e6 in a QE number of peptides, so 1 million peptides that we can actually analyze at a given time in a given scan. So if you've got a nice representation of your 3 peptides amongst those million ions, then you can get really good measurements for their precursor scans. But realistically, if there are fewer and fewer of one particular population, you might not have any of those ions actually in that 1 in a million HCD cell for being able to collect ions. So if we're trying to collect 1 million ions, and we're actually measuring something that is, 3 or 4 orders of magnitude, 5 or 6 orders of magnitude down, there might just not even be any measurable ions associated with the peptide that you're interested in. And all of a sudden your signal for that particular peptide disappears. But this is actually one of the reasons why techniques like boxcar work, is that with boxcar, you're actually using the quadrupole up front to filter out ions that are not associated with the region that you want to look at the precursor space for. So if you only measure, in this particular case, the precursor mass around the orange ion, then you're going to definitely see measurements for that orange ion, which is why boxcar is so effective at being able to see good precursor signals for peptides. DIA is essentially the same thing, just a much earlier approach. So the advantage of this is that, as opposed to just getting an accurate precursor mass with boxcar, you actually get fragment ions associated with those peptides. So we're just doing fragmentation in the HCD cell and measuring all the fragment ions for those selected ions. So in a sense, boxcar and DIA are actually really similar concepts. It's just whether you're collecting precursors or fragment ions associated with those peptides. Okay, so this is why you would want to do DIA versus a DDA. There are many different sort of reasons for this, but why would you want to do it over PRMs or SRMs? And a lot of that actually has to do with the sort of workflow in which you're analyzing PRMs. In this particular case. So Gabriella, actually boxcar you wouldn't want to use to build a library, because you actually need a library to analyze boxcar data. Because you aren't necessarily collecting fragment ions, you only have precursor ions. So you don't necessarily know what the particular peptide sequence is, you just know what the precursor signal is, because that's all you have time to do. Now there are combinations of boxcar plus DDA or boxcar plus DIA that kind of get that make the boxcar be able to analyze data in the same, or interpret the data that is collected for quantitative work in the same experiment. But those are more complicated types of data acquisition. But if we're thinking about PRMs, one of the things that we have to do with PRMs is we have to think and sit down and think about what peptides are we going to select, because we only got 100 peptides or 300 peptides that we can actually measure at any given time point. So we have to think about which peptides we're going to measure. We have to select those particular peptides. We have to figure out when they're going to elute, and we're going to have to come up with which transitions represent those peptides best. And only then can we start acquiring data for those peptides. And so oftentimes what we do is we acquire a preliminary experiment, and then we say, actually, we needed to measure these other peptides at the same time because they belong to a different pathway that is also really important. And then we got to go back and select some new peptides and do some more scheduling and some more refinement. It's actually a long process to be able to build a robust PRM or SRM assay. It actually takes months to be able to make these things very robust and very accurate. And so good target experiments take a ton of time. because of this sort of got to think a lot before you actually acquire data. With DIA, we actually have this interesting alternate problem, which is that that we can actually acquire data up front. We don't even need to think about which peptides we are interested in monitoring, because we're collecting them across all of the peptides from 400 to 1200 or 400 to 1000 MZ. And then we can come up with our hypotheses. We can do our peptide selection and chromatogram extraction. Instead of doing these refinements, we can actually just think of it as extraction out of the DIA data and this transition refinement out of the DIA data after it's been acquired to decide which transitions are actually useful for making those measurements of those particular peptides. And then we can go back and we can circle around on the data analysis rather than trying to circle around and have to reacquire new data after one after another. Okay, so now that we've started to think about what DIA data is and how sort of we can roughly think about it from one experiment to another experiment, let's look at how we actually do some analysis of this type of data. And we typically are analyzing this data with libraries, but let's reason why we actually need to do this. So with DIA, this issue of peptide selection is really important. So you could think about going from the most broad, you could actually look at all possible tryptic peptides associated with all possible proteins in your proteome. That would be sort of the most broad DDA style approach, all the peptides in your FASTA database. Down to, you can do the predicting for all the hyper-responding peptides. You can look at stuff that you've already seen in DDA libraries. You can build DIA-specific libraries, which we'll talk about at the very end. Or you could actually just look at peptides that are identified by DDA in your specific samples, which is maybe the most limiting approach. So why can't we analyze all tryptic peptides in the proteome? And let me give you kind of an example. Here is a protein that is a transcription factor called Cas-Z1. And these are the transition responses. And again, this is on the log scale. So this is the actual responses in signal from the first peptide, the most abundant or most highly signaled peptide, and the lowest peptide. And this has to do with response in the mass spectrometer. Certain types of peptides ionize well, certain peptides release from the protein with trypsin well, and other peptides don't necessarily release as well. And so what we're looking at in this particular sample, or this particular protein, Cas-Z1, is 3 orders of magnitude, 1 to 1,000, or in this case, even more than that. So it's 2.5 orders of magnitude in this case. Response between the best responder and the worst responder. And this is actually really common due to ionization. If we look across, in this particular case, I was looking at almost 1,000 different proteins in terms of their best response and worst response in equimolar proteins that were, in this particular case, synthesized for this very problem. So we knew we had the full-length protein and we had equimolar peptides for every single one of those peptides on column, or at least available to trypsin at that point. And we see 3 orders of magnitude is actually the average of 3 to 4 orders of magnitude is the average separation between the top responder and the worst responder. So realistically, in a DIA experiment, we're going to see the first couple of peptides, the most easy to see peptides, and all the peptides at the bottom are just not even worth thinking about. They're not even worth considering because they are such poor responders. So that's why you actually, when you're looking at a peptide sequence, you actually are only seeing a handful of peptides across that protein, not all the peptides across that protein. Okay, so that kind of gives you some intuition for why you can't look at all tryptic peptides. But so most of the way we think about this is from thinking about libraries, specifically around DDA spectral libraries, because that's sort of the most common way to think about this type of peptide. So one of the ways in which we can do this is, so again, just thinking about what we talked about earlier about extracting out fragment ions, where we're looking at the library spectrum versus actual spectra. The way in which we try to make this type of detection is we monitor over time. We do this sort of extraction of all of the fragment ions out over time. So this is, again, all those different peaks. And each time point, we actually score the library spectrum versus the MSMS spectrum that we've acquired, or the fragment ions that we expect to see for that specific peptide. And so we expect that score to, be around a baseline, and then at a certain point spike up. And we hope that at that certain point where it's spiked up, that's actually an indicator of the particular peptide. So if you are interested, for example, Simi, in analyzing triptych peptides and semi-triptych peptides, so very complicated types of peptides, or for example, phosphopeptides or unusual types of modifications, those circumstances you would need to basically build a library specific for those types of peptides and look for those specific peptides as you're going. Okay. So how do we analyze this type of data? Specifically, a lot of you guys have seen sort of target decoy analysis for computing false discovery rate, where you actually have, you search for a forward FASTA database as your normal FASTA database, and you've got a reverse FASTA database that you search at the same time. And the advantage of searching this as a combination is that you can say, well, you're not really sure about any particular match to the forward database, but you can know that any match to the reverse database is definitely wrong because those peptides don't exist in the real world. So that kind of gives you a way to say, what do bad peptides look like? What are the scores for bad peptides? And how far away, how much better are the scores for the forward peptides than than these reverse peptides, as a way to judge how confident you are in that type of measurement. You could do the same type of thing with library spectra, actually, where you've got sort of target spectra, and then you can construct decoy spectra. And the way we construct these decoy spectra is we take the sequence and then we reverse the sequence except for the termini. So we keep the, in this particular case, the V and of the vape peptide and the K the same, and then we reverse all of the sequence in the middle, and then we keep the same ion intensities, but we move the masses around and potentially the mass errors around for those particular fragments. So you can see that the intensities are the same for the ones that I've selected out, but their masses are a little bit different. That helps us be able to say this is a decoy rather than a normal spectrum. Okay. So this is one approach to analyzing data. This is called the peptide-centric approach, where we're extracting out all of the fragment ions that may be associated with that over time. And there are some great advantages for it, but I want to give you another idea for how you could also interpret this type of data. And that's using a method called DI-Umpire, and there's a bunch of other types of methods that try to do the same type of thing. But the idea is basically they try to make pseudospectra from DIA. That's where you've got DIA data over time, and you want to extract out what a specific fragment ion, what a specific fragments are associated with an individual precursor are. And so they look at the sort of extraction for a particular precursor that they say that is of interest, and then they look for any fragment ions that have that same retention time characteristic, the same rise and fall as that precursor peak. Now this runs into a lot of the same problems because it requires that you actually have a precursor signal that is accurate and representative of the shape of the peak, which we talked about earlier as not being necessarily a perfect scenario and some of the things that DIA can resolve. But it is an approach where we say these particular precursors can be assigned to these specific fragment ions, and that lets us compute a pseudo spectra of fragment ions that have the same rise and fall as that individual precursor. And then we can send those guys through a normal DDA-like platform where we're analyzing data with Mascot or with Proteome Discover or something like that. Okay, so what are the advantages for this sort of spectrum-centric approach is that we can use it like normal DDA-based methods. And so here we can take our DIA experiment, we can condense it down into like 30 to 50,000 MSMS spectra that are pseudo-MS spectra. And then we can check each one of those against a protein sequence database, and we make a peptide spectrum match, or a PSM, as we talked about earlier, based off of each spectrum, right? Okay, so this lets us say, okay, we've got 50,000 spectra that we're interested in, and so now we're trying to make 50,000 assignments for peptides in that experiment. The alternative approach here would be like taking all of the peptides in a FASTA database, say that 1 to 2 million peptides of interest, extracting out signal for all of those different peptides, and then determining what evidence is there for that detection. So this is interesting because it gives you the ability to say, well, there was no evidence for this particular peptide. That's something that you cannot do with a spectrum-centric analysis method. But it does have this major caveat where we're actually analyzing, again, in the top case, we are looking at 50,000 spectra. In the bottom case, we're actually looking at 1 to 2 million peptides. So what we're actually interested in is trying to we're essentially asking 50 times more questions, which means we need to false discovery correct against a lot more types of peptides at the same analysis platform or same analysis set. Okay, so how do we think about building these types of libraries? Because you want to do this across a lot of different fragment ions, a lot of different peptides, across many different types of experiments. And so one way you can kind of think about this is using stable isotope labeled peptides. These can be any stable isotope labeled peptides or any peptides that you know that are not in your sample. So we talk oftentimes about IRT peptides, but you can also use other types of peptides. So for example, Pierce has a retention time standard that I frequently use. You can use BSA peptides. Honestly, you can use peptides that are actually endogenous in your sample just normally. But if you know an actual retention time for those particular peptides and you can map it to some index retention time, and again, here I'm using IRT as my index retention time, but it doesn't actually have to be IRTs. IRT is kind of like a dimensionless place that has no necessarily bearing in the real world. Like you can get negative IRT retention times, which makes no sense in the real world. So I can extract out those blue peptides out of my DDA run when I've injected them into, or when I put them into my sample and collected DDA. And then I can say for all of my other peptides that I've measured, I can map those to a specific point in that retention time line. So now I have a specific retention time IRT for all of those different peptides that I'm interested in monitoring. And so if I've got another type of peptide or another type of DDA run, maybe another fraction or something like that of the same type of sample or a different sample entirely, I can run a different DDA experiment, and it doesn't matter if those IRT peptides move around. They actually still give me enough anchors to be able to map to that same type of line. Now, Hugo asks, how many peptides do I need for our standard? Honestly, I think that the IRT standard is around 10 peptides. The PRTC is around 12 peptides. Something in that range is just fine. Digested, as I said, just taking enolase and digesting it, or taking albumin and digesting it, and then putting it into your sample at a certain small ratio is enough as well. So that's another good way to try to deal with this, as long as those peptides are not likely to be in your specific sample. So this is one way to think of it, and then you can map those retention times from one run to the next run. The other way to think of this is once you've actually mapped all those red peptides in that first DDA run to IRTs, now all of a sudden you actually have IRT measurements for all those peptides. So anytime you see those peptides again, they become your IRT experiment. So Hugo, thinking about this, you can actually use these now peptides, now that you've put IRT, now that you put heavy stable isotope labeled peptides into one sample, you can use any peptide that is assigned in that sample as now an IRT mapper. And so one type of approach that we published a couple of years ago takes those peptides and monitor it in one particular sample, and we map those to IRT samples. Now we can go back to all of our old data and we can say, well, we've got a bunch of the peptides because they're all within the same sort of framework. If I analyze, for example, in that red experiment, if I analyze and identify 5,000 peptides, now I have 5,000 anchors. And again, because I only need 10 of them in any given sample to be able to draw that line, I can now look at a green sample, totally doesn't have any IRT sample peptides in it, no stable isotope labeled peptides in it at all, but it has some of the red peptides in it. And I can look at a purple sample, and I can look at a blue sample, and I can combine a ton of data. So in this experiment that we did at this point 4 or 5 years ago now, we actually were looking at a swath of 1,000 different DDA runs that were acquired quite a long time ago, actually. This is basically our ability to take and mine all of those runs based off of a single DDA run that we ran, because we were able to assign anchors to all of those DDA, all the peptides identified on that single run, and then map all those new peptides into our new set of samples. So this is a great way to mine all the old data that exists in your lab, on your system, on your setups. That said, there are a lot of different other public available libraries. You can go to Swath Atlas, download some libraries. You can go to NIST and download libraries. That library that we talked about earlier, where we combined a bunch of data together, that was actually a phospholipeptide library linked into this library called Phosphopedia that we put up. So that's another great library source to be able to get access to for DIA or PRM types of experiments. So again, all of these are great sources. There's a growing list of these library sources, and I think you can definitely be excited about all those different types of sources, but be careful with them because they're not always ideal. You need to make sure that they're good for your type of experiment. So there are lots of different types of tools that are good for this type of experiment. We've talked about a bunch of these, like Skyline. There are also other types of, for example, retention time-based filtering approaches like OpenSwath or Spectronaut or PeakView, some spectrum-centric approaches like DIA Umpire or PLGS, some RT-agnostic approaches like Encyclopedia or Scaffold DIA, MS Split DIA, Pecan. There are tools that now split between these that take multiple types of approaches. So DIA Neural Network, DINN, uses kind of a retention time-based filtering and then kind of thinks about it from a spectrum-centric perspective. There's Direct DIA from Spectronaut that kind of is a spectrum-centric tool as well. So there's like lots of different ways in which you can type of work with this, but they all they fall into these different brackets, but they all have different good and bad aspects to them. Again, the spectrum-centric ones all require precursors. The retention time filtering-based ones require really accurate retention times to be useful. The retention time agnostic ones are sort of more free, but at the same time become a little bit harder to judge FDR with. So there are lots of different kind of great advantages and disadvantages of each of these different quadrants. Okay, so why would we want to do this? One thing that we've noticed is that if we take a particular data set where we analyze it with PRM and we come up with good ratios for what a peptide specifically should be, we can be really confident that that type of measurement, that quantitative measurement is the right measurement. We can compare that with DIA methods. In this particular case, I've got 3 different DIA methods where I've got normal DIA, variable width DIA, overlap DIA, and we've got a DDA-based method. And one of the things that we find is that more of the time, we see that the DIA-based methods, they identify about the same number of peptides usually in this particular case, but they quantify much more accurately for DIA. So we get a ratio of what we measured versus what we expected to be as close to 1-to-1 as possible, whereas there's a little bit more scatter with the DDA measurement. And so one of the things that we see is that we see lots of these circumstances where if we look at sort of the width of that box over time, we find that the DDA actually just tends to introduce a lot more noise into the types of measurements that we're making. And one of the reasons for this has to do with the fact that with DDA, you are extremely reliant on getting an accurate monoisotopic measurement, +1 measurement, +2 measurement. If any one of those measurements is interfered with, then the overall signal that you're trying to work with is actually interfered with. So one of the advantages of DIA is that you can almost always get 3 good transitions out of any given peptide measurement. So in this particular case, over 80% of the detections that we're making here in this experiment have over 3 transitions. And because of that, because we've got 3 good measurements for those individual peptides, we actually are looking at CVs well below 20% on the vast majority of these cases. So we can be really comfortable that we're making good measurements in these cases because we can always throw out transitions. We're not reliant on being able to make measurements of we're not reliant on accurate measurements for everything that we measure. So for example, with DDA, you need the monoisotopic, you need that +1, the +2 to be able to integrate that peptide well. Here, we only need 3 transitions to work. Okay, so what are our potential pitfalls? I'm just going to go through this really quickly. One of those particular pitfalls is that oftentimes when we're looking at a particular peptide, we actually can see signal for that peptide that corresponds to that particular peptide from multiple different sources. What's going on in this particular case is I'm actually looking at a phosphopeptide experiment where a phosphate is actually being moved from one peptide to the other peptide, same sequence, but just different localizations of that peptide. And because there are different localizations, they move from one side, the retention time shifts considerably, and the relative abundance of the different fragment ions is a little bit different. Well, you get kind of 2 different sets of measurements. So what's going on here is if we're measuring, for example, these 2 CDK peptides, we've got a phosphopeptide where the tyrosine is phosphorylated down here and the threonine is phosphorylated up here. They have exactly the same parent masses, and the retention times may be a little bit different. But realistically, the only way that you can tell the difference between these is by using those specific fragment ions. So here, for example, we've got an isobaric peptide. So this is exactly the same mass, right? So both of these peptides have exactly the same mass, but they share a lot of the same orange peptides here. But there are some yellow peptide fragment ions and some blue fragment ions that are specific to one form versus the other, and that helps give you a chance to tell the sort of difference between these apart. And there are lots of different other types of modifications that can kind of cause different types of problems in these circumstances that you can look at. So, for example, deamidation or oxidation all can sometimes exist within the same window. So, for example, if I go here, I can, and I'm just going to skip a little slide just to make this a little bit more straightforward. So if I'm interested in analyzing this particular peptide and I'm looking at the modified, the sort of unmodified form and the oxidized modified form, they have very similar precursors. And if you look at all of the Y-ion fragment ions, they're all going to be exactly the same between these guys. So these red ions are going to be exactly the same between the only thing that you're going to be able to tell the difference on are these different B ions. And so when you look at these precursors, they actually exist within that same 24, 25 MZ wide isolation window. And so you're co-fragmenting them at the same time. And so if you actually try to do extraction of these particular peptides out at the same time, you get essentially the same signals. And so you might actually end up mistaking one for the other. And so this becomes a potential pitfall. So this is one of the reasons why we don't necessarily recommend searching for modifications in a lot of these types of searches. Or if you're searching for modifications, be very sort of savvy about what your precursor isolation window widths are and that sort of thing. Okay. So let's see, this is a good point to start. So Hugo asks, should we discard less than 3 transitions with DIA quant? Oftentimes, I usually require 3 transitions. I think this is a really good way to go. But you can work with fewer transitions. You just have to know that your signal, your measurements are actually just not as good or not as accurate. So typically, I require 3. Oftentimes, I actually will require even more than that. And Lima asks, for IRTs, could I use a digestive standard of enolase? Absolutely. Yeah. So that's what I'm saying is, as long as it's not a protein that you expect to see in your sample and you're not trying to measure in your sample, you can absolutely use it. Also, if you know a retention time standard for any given peptide in your sample, you can use that as the type of measurement. Sumit asks, can you use something like iTrack with DIA? Reasonably, you cannot, actually. iTrack and DIA are pretty much not You can use something like complementary TMT potentially, theoretically, but it's just not a very, it's not likely to be, there's a lot of research that needs to go into that type of work before you actually could do something like that. You definitely can't use something like normal eye track because of all of the quantitative information is essentially lost because you're actively seeing co-eluting isobaric peptides at the same time. Okay, so let's talk about briefly best practices for, before we get there, Leanne, am I saying that DIA is not great for looking at PTMs? I think actually DIA is one of the best ways to look at PTMs, but you have to be really savvy about what types of PTMs you're measuring, and you have to be able to use localization search engines. And we published some of those as well. So with an approach called Thesaurus that we are really excited about. Where we actually see a lot of faults of doing DDA analysis. In fact, from our perspective, we think that you can't really analyze, you can't get robust information about phospholipeptides from DDA based off of the information that we've learned from analyzing phospholipeptides with DIA. So we think that the DIA is a perfect tool for being able to analyze phospholipeptides, but you have to be smart about how you analyze them. You have to use the right software. You have to think about it a lot. Okay, so best practices for setting up DIA, and then we're going to talk about building DIA-only library data sets because they kind of go hand in hand. Because at this point, from my perspective, actually, I have stopped acquiring DDA-based libraries. Most of my work now uses DIA-only libraries because I find it to be just as accurate and considerably faster for the work that I am interested in doing. So to understand how you set up a DIA experiment, you actually have to go back to think about how an instrument actually works. A lot of the DIA experiments were originally designed for TOFs because TOFs actually have a lot of the same types of characteristics as triple quads. So one kind of advantage here is that that basically you rather than having a QQQ, you actually have a QQ TOF, right? And so here, essentially, all you're doing is you're taking peptides that you're interested in. You select out a specific mass over charge range. You fragment those ions in the middle quadrupole, and then you push them into the TOF. And so the TOF pushes them down, and they hit the detector. And the advantage of this is that you get measurements that are very, very fast. So thousands and thousands of pushes per second. And the other advantage of this is you get real scans, real profile scans for every different type of peptide. So you're always going to see signal after you've analyzed enough of these pushes. But one kind of disadvantage of this approach is that the pushes happen so fast that that you actually aren't looking at that many ions at any given time point. So if you're thinking about what you're looking at a millihertz kind of scan, so if you're looking at 1 in 1,000 1 over 1,000 seconds to accumulate ions and put them into the TOF, that's not very many ions, realistically. Okay, so a lot of thinking about the original sort of Swath paper that was interested in analyzing all peptides from 400 to 425 across this mass range. So they built out 32 windows that were 25 MZ wide. One of the things that they were pretty clever about early on was thinking about the sort of distribution of peptides that they're actually being able to see. Most peptides actually exist in this 400 to 1,000 range here, and there are fewer and fewer peptides up above that. So you can use essentially what are called variable width windows across that time point. And so the earliest approaches use 32 windows and then showed the variable windows where you have wider windows, the higher MZ range where you have fewer peptides that are co-eluting at the same time, and narrower windows at the low range work pretty well. But 32 windows, because the TOF can scan so fast, they thought that you might as well try to sort of push this. So 30 windows became 40 windows, became 60 windows, became 120 windows. And so this is kind of like the current standard now, where your minimum window size for the windows at the middle are around 5 MZ wide windows. And they get out to around 40 MZ at the top range. But that best range is in that 5 MZ range. So an approach that people have been doing, remember how I talked about how transmission efficiency on the window edges is not as good. One way to try to deal with that is by just tacking on margins onto the side of those windows. So this is, I think, a very good way of analyzing data using a TOF. Now, I want to contrast. So this is sort of the common practice that has been pushed by, for example, SciEx. And I think this is a great way to try to do this type of work. Okay, so that said, there are very different constraints when you're thinking about this with an Orbitrap. So if you're analyzing data with an Orbitrap and trying to do DIA here, you end up with an Orbitrap, which is actually a relatively slow mass measuring device. So instead of being able to analyze thousands and thousands of scans per second, you're actually only analyze maybe 10 to 20 scans per second.
Brian,
so
there's a question about TOF. So how much scan time should I set, or at least a reasonable time? 0.3, 0.5, 1 second?
Oh, again, that really depends a lot on the specific TOF that you're actually working with. That one, I think, is worth talking to the specific vendor. So if you're working with SciEx or Agilent, you should talk to the vendor about how you should configure the instrument on those specific TOFs. Again, remember the kind of constraints that we've got where you have a need to be able to make the same measurement every 2.5 or 3 seconds wide. So Sean asks, would a TIMS TOF setup be similar to a SciEx TOF? And actually, it's very different. A TIMS TOF, because of the constraints of having the ion mobility cell up front, you actually have very different ways of acquiring that data. And so all of the TIMS TOF DIA approaches so far have actually gone back to the original 25 MZ wide windows across the same range approach because of the need of making everything work within the ion mobility space of the TIMS device. Okay, so again, an Orbitrap is a relatively slow device. So if we're thinking about trying to make measurements across that same type of window, you know, at best, we're looking at 25 windows in that same range. We are actually looking at very, very wide variable width windows. So we're talking about 20 MZ wide windows in the best part and up to 150 MZ wide windows up at the long part. So this actually is kind of a terrible way to use a TOF, an Orbitrap, is to basically take the ideas that were developed for TOF instruments and apply them here. We really need to develop new types of ideas here. Okay, so there are some pretty big advantages in trying to do this with a TOF. And I want to just sort of brush through these really quickly to give you kind of an idea. One of them is that we really kind of can think about limiting the mass range a little bit more. So historically, we've only been looking at from 400 to 1200. And really right here, actually, we could focus in on this range from 400 to 1000. And we have some data that actually backs up this idea of saying that we get the most number of detections from this 400 to 1000 different window set for baking these types of measurements with an Orbitrap, and that range seems like it's the best type of range for our types of experiments. But if you're looking at phosphopeptides, you actually kind of would want to have a wider window or a different type of window. So maybe you would consider from 500 to 1100, for example. Another approach that I think is a really powerful approach is essentially an algorithmic way of doubling your resolution with the instrument. And so if we think about this type of approach where we've got precursor scans and fragment ion scans one after another, and they've got window boundaries as shown here, well, we can think about, well, what if we shifted over every other scan, every other set of scans for cycle by 50%. And this goes back to that first question about, well, whether we could analyze overlapping scan windows, and that we can actually use this to our advantage. So if we have overlapping scan windows, or in this case, we like to call it staggered windows. So if you're looking at an MS2, for example, here, all of those ions in black for that given MS2, here is a particular window that you're looking at. And if you look at the windows immediately before and immediately after, some ions are going to be present in both of those cases. So these red ions, you aren't going to be able to say much about. But if you find ions only in the early part, so in this particular case, if we're looking at our center window is from 600 to 624, and our previous window set is from 588 to 612, we see that there are ions in green. And these ions in green, we can say, are definitely only in the left side and not in the right side. And so we can actually assign those fragment ions only to a scan that belongs from this 600 to 612 range, because they have to exist in that first window and not at all in the second window. And so we can do the same thing on the other side, where any ions that are associated with the previous and next scans on the right side can be associated with a new spectrum, a pseudospectrum that is associated only with the range from 612 to 624. And so this is a really great way of simplifying our experiments and being able to essentially increase the speed at which we're acquiring data by 2 times without having to do any actual adjustments to the instrument. I mean, think about this. If Thermo were to come out and say, we've got an instrument that collects Orbitrap scans twice as fast, you'd be like, wow, that's amazing. Here we're actually able to do that algorithmically without even having to change the instrument. You can do this on any type of instrument that Thermo's made for this. That said, staggering is incompatible with this idea about putting margins on the side. So we have to come up with a different strategy for dealing with that type of thing. And this is where we think about what we call forbidden zones. Forbidden zones take advantage of the fact that peptides are made of the same stuff. They're all made of hydrogen, carbon, nitrogen, oxygen, sulfur. And because they're made of the same stuff, they only can have certain precursor masses. So there are certain types of masses that they just cannot take because they are not made of weird atomic, different types of heavy metals or different types of other atoms. So those spaces between where you expect to see peptides are where we can put what we call optimal boundaries. And these optimal boundaries are reasonably wide. So for example, if I look at, and they seem relatively straightforward. So when I think of the boundaries that I would look at for Swissprot and map those to the boundaries that are where I see peptides, and looking at all the peptides at Swissprot and then all the peptides in the pan-human library, they actually overlap quite a bit. And they fall in these kind of peaks where you expect, so any peptide that has a +1, a +2, or +3 charge falls in this particular high peak. +4s are in the shoulder, +3s are in this peak here, +2s and 4s are in this peak here, +3s are here, +4s are in the shoulder. And again, you get to the next monoisotopic peak where you get +1s again. So this is where you expect to find peptides, where you see these signals. So it's very easy just to stick those boundaries in a region where you don't expect to see peptides. That said, other types of peptides have different forbidden zones based off of their chemical characteristics. So you have to compute these separately. So for example, phosphopeptides have a slightly shifted forbidden zone that we've seen based off of different libraries, and other types of modifications have different forbidden zones. So if you try to do this type of work, it's worth trying to think about the math for where you would draw these boundaries. Okay. A final sort of approach that we like to take is this sort of idea of gas phase fractionation. That's where, rather than trying to analyze an entire acquisition or entire sample within one acquisition, so from 400 to 1,000, we're actually interested in just saying, well, what if we injected it twice? And instead of trying to just do it twice like technical replicates like you would do for DDA, why don't we just make the first measurement from 400 to 700 and the second measurement from 700 to 1,000. This gives us much narrower windows and gives us better isolation and better characteristics of being able to identify peptides. So if you have the ability to spend multiple acquisitions, we find that this is a very accurate, very good way of dealing with this, as described originally by Dave Goodlett's lab in the early in 2008, 2009, and again, sort of reused in this particular case for modern DIA in Sonia Ting's paper in 2017. And we've got on our website, and again, you have access to these slides, so you don't need to write down these crazy long URLs, but we've got lists of parameters that we recommend for how you would set up these types of experiments based on, as well as the window widths, based on the sort of constraints that we've talked about so far. So what you're going to set your AGC targets for and your max ion inject times and those types of things that you would want. Okay. Again, I would also recommend referring to this paper that I put into the Google Doc, but also just to read through it to kind of get a refresher on this type of stuff. We've also got a quick start guide for how to deal with this. Again, that same recommendations for DIA settings on Orbitraps. So in the last just sort of couple minutes, I want to talk very briefly about this idea about building libraries for DIA-only data sets. And this is something that's actually been really important in our lab that we've been moving forward on. And the reason why is because DDA libraries are, they're very expensive to make, right? So if you're interested in analyzing a specific sample, say HeLa or MCF7 cells or something like that, or yeast or E. coli, you really need to acquire tens of samples tens of injections of fractionation to be able to analyze that, to be able to representative library for that particular sample. And one of the things that actually explodes if you're trying to monitor all of or detect all of the possible peptides for that organism. These experiments take a lot of effort, a lot of time. And it gets even worse, actually. If you're trying to look at the overlap between different types of fractionation methods, they actually identify very different types of peptides. So if you're using SEX or high pH reverse phase to do your fractionation to build libraries, you identify very different types of peptides. So in those particular cases, because those different types of peptides identify, different blocks, you actually kind of need to use multiple types of fractionation methods to build those peptides. The other aspect of this is that collision energy optimizations are actually a little bit off. So for example, as we talked about originally, DIA is not really perfectly set up for being able to make collision energy or the fragmentation parameters for DIA are not exactly the same as for DDA. And as a result, if you're looking at Pearson correlation, as I'm showing at the bottom, most peptides have good Pearson correlation if they're +2 peptides, but +3 peptides tend to do a little bit worse, and +4 peptides tend to have very bad correlation relative to what you see with DDA. I think the final and most kind of weirdest aspect of this is that you actually see matrix effects caused by fragmentation, by sort of fractionation methods. So if you're analyzing a sample with a much lower matrix than your actual samples are, then you get retention time shifts because there are different types of peptides that any peptide you're interested in is going to be interacting with on that column. So you end up with sort of these time shifts in weird areas, and you get shifting of different peptides backwards and forwards because of the fact that they have a simplified matrix associated with them. So that even if you're using lots of different fractionation methods, you still get inaccurate retention times because of this. So we've actually been adopting this kind of approach of trying to do library prediction. In this particular case, we're using PROSIT quite a lot. And this is a tool that we recommend. And we've developed tools and workflows around this in sort of inside of Encyclopedia, where there are mechanisms to take a FASTA database and convert it into a PROSIT input so that you can stick that up on a PROSIT website, and then analyze those data, bring those back into Encyclopedia, build libraries for them for DIA, and be able to analyze data with them. And we find that the predictions are actually really shockingly accurate. So for example, this is a particular peptide. I pulled this one at random, where you can see the fragmentation patterns of this particular peptide are almost exactly on. They're not always perfect, but they are almost always like within a very reasonable error margin. And the retention time prediction based off of the deep learning model in PROSIT is off in this particular case by maybe 1.5 minutes, but that's actually about as accurate as a DDA experiment would be with fractionation. So to kind of give you an idea, basically PROSIT is predicting libraries that are as accurate as DDA-based libraries, is kind of our take-home based message on this. But still, when we're analyzing data with these DDA libraries, we actually still see more peptides, more proteins than we do with these predicted libraries. And again, these here we're making a combined SEX, HPH, high pH reverse phase, DDA-based library. So in analyzing yeast, we're looking at 16 fractions here, and analyzing HeLa, we're looking at 39 fractions. We're identifying a little bit more than we do with the predicted library. And again, this has to do with the fact that we're analyzing all the tryptic peptides in the FASTA database. And there's just too many peptides for us to look at. And so one of those aspects is We really need to look at as small a database as possible. Again, don't consider PTMs unless your experiment is about PTMs and you have enriched for those specific PTMs. But we've started to work on this approach of trying to make it a little bit easier to analyze this type of data, where instead of trying to think about gas phase fractionation for 2 acquisitions, we actually have increased this a little bit more, where we're actually doing 6 different fractions associated over time. So we're doing 1 fraction acquisition, a second acquisition, so 6 injections for this sample, in which we can analyze the sample with a huge degree of accuracy, with basically, essentially, very narrow precursor isolation windows. And so this is kind of like the new workflow that we've been using, where we take these sort of samples that we acquire from our different pools. So we're basically taking a pool of this yellow sample from our blue samples and our orange samples. We're ejecting that yellow sample 6 times using gas phase fractionation. We're searching that using the PROSIT predicted library, and then we're making here a library that is associated with all of the possible peptides that we can see in this sample. So now we've identified all the peptides that we're confident in seeing in either the orange or the blue samples. And now we could just take our quantitative samples and then look those guys up very accurately. And the advantage of this approach is that we actually get extremely accurate retention times in this approach called the chromatogram library approach. So instead of looking at plus or minus 5-minute windows, we're talking about plus or minus 20-second windows, because we know exactly where that retention time should be. And our fragmentation, because it's acquired with DIA, we're actually getting really good DIA-based measurements here. So finally, we see with this type of approach, we actually are getting about the same number of detections as we do with these very large libraries. So for example, if we can use this empirical correction, this chromatogram library and correction, to improve any type of detection approach. So in this particular case, we're looking at here, if we're looking at the DDA libraries, we can actually build libraries that actually help us identify more peptides. And then we can do the same thing with these predicted libraries. But I think the biggest advantage here is that now we essentially make the empirical corrected DDA libraries essentially the same as the empirical corrected predicted libraries. So this enables us to build like a DIA only workflow that it allows us to be able to identify every type of peptide. So with this, I'd just like to close with a couple of recommendations. DIA is a fantastic tool. It's not the end-all be-all for every type of experiment, but if you just need a quick answer and you need kind of like, you've got a really simple matrix, then DDA can be a really great tool. But there are different types of approaches that I would recommend, for example, using gas phase fractionation if you have only a handful of samples. If you've got a lot of samples, then I recommend building these gas phase fractionation libraries, these chromatogram libraries, again, using predicted libraries, and then using DIA, a single injection DIA to analyze your quantitative samples. PRM is sort of the same workflow, or sorry, with PTMs, it's the same workflow, except there I would highly recommend using a DDA-based library because the prediction algorithms aren't very good right now for modified peptides. So with that, I'd like to finish, and I'd be happy to stick around in the break. I know we've gone over a little bit, so I'll stick around in the time between Lindsay's art session and then Lindsay's session and answer a bunch of these questions as we go.
Brian, thank you so very much. We have a virtual round of applause for Brian. Thank you for this comprehensive overview. There are quite a few questions left in the Q&A, and there's also one question for you in the chat at the very bottom of the chat as well. So for everyone else, we have a break until 12:45, so we can use this time to go over questions or just stretch your legs, and we will be back at 12:45 Eastern for Lindsay's presentation.
Great. So I'll just kind of like go through these a bit. So for example, the one issue with the staggered window scheme, that's the overlapping window scheme that I talked a little bit about. Let's see back here. So this approach here, I said that this approach here isn't compatible with window margins. It's also not compatible with variable width windows too. So that is kind of a limitation. But there are kind of other interesting ways in which you would want to think about this from a TOF perspective. So David Schmidt brings up a really interesting point about what about scanning swath methods? That's where you actually take the quadrupole and you sort of scan it across time. And actually, Waters came up with the first implementation of this called Sonar. So Sonar came out a couple of years before scanning swath they're essentially the same type of approach where you're scanning the quadrupole over a specific window and you're essentially acquiring micro scans at each of those different windows, and you can use linear algebra to be able to deconvolute those together. It's actually a lot of the same type of math that we're doing here with these fixed scans with the Orbitrap. You can't do a scanning Orbitrap essentially because the Orbitrap is too slow. So those are great questions. So when you're interested, Guthrie asks about using SIL peptides or stable isotope labeled peptides for doing DIA analysis with Encyclopedia. So right now the only way to do to integrate those with Encyclopedia is to actually have them in your library as well. So you can definitely do the integrations for them, but you treat them as independent detections and you interpret them on their own. So for example, if you've got your normal library, you can add in the stable isotope labeled library or for those specific peptides at least. So that's a fantastic question. There's actually new tooling in the new 1.2.2 to be able to deal with that type of library addition. Okay, so can I elaborate a little bit more on the GPF strategy for building libraries? And I want to show you this slide here again. Okay, so here again, so we've got 30 samples. Say we've got 30 samples and we've got 15 blue samples, 15 orange samples. We want to analyze these together and we want to build a library for them. So what I will do in this type of experiment is for these 30 samples, I will take a sub-aliquot off the top of each one. And oftentimes you can get a microliter out of the bottom of each well anyway. So this is free sample when you think about it. So I will take a sub-aliquot off of the bottom of each one of those, pool them together into a single pool. That's that yellow sample. And then this yellow sample, this pool, I will analyze this with a different framework. So the orange and blue samples, I will analyze those for quantitation. And the yellow sample, I will analyze for detection. I want to detect every possible peptide in that sample as accurately as possible. So I do this sort of 6 gas phase fractionation method where I acquire 2 MZ wide windows across the entire window range, 400 to 1,000. This is basically like doing PRM, right? PRM isolation is about 2 MZ wide windows, but it's PRM for every peptide in the proteome. You can't do this for every sample, right? That would be way too much work to do 6 injections for every sample. But you can do it for a pool very efficiently. So in this particular case, I would acquire my 30 orange and blue samples and my 6 gas phase fractions for my pool. And so now I've got 6 injections of PRM quality data. I detect all of the possible peptides out of this data set. So again, 2 MZ wide isolation means that this is as good as DDA based isolation. So you're seeing as those types of peptides in the same so you're seeing them with the same type of isolation that you would with DDA, except you're actually able to see the apex of the peak, not just the shoulder of the peak for collecting the MS2s. They're just much deeper. Typically in these gas phase fractionation libraries, we can identify tens of thousands of so oftentimes we can see up to 100,000 different peptides that we can confidently identify. And once we found them in the pool, well, now we know the exact fragmentation patterns and the exact retention time standards for those specific peptides. We can map those to the rest of the experiment. So for example, I can say, well, I found this peptide that I'm interested in. I know exactly where it is in time and space, and I can go look it up with the right tools. So we've tried to make this workflow very easy to use in Encyclopedia, but you can use this with other types of methods too. We've done it with Skyline, for example, before. Okay, great. So Charlie Yang asked the question about IRT peptides. IRT peptides are just a trademarked peptide type that fit within index retention time. These are peptides that are produced by a specific company, Biognosys. So they're sold by that company, and that's fine. But any type of peptide works here. So pure retention time standards, PRTCs, are essentially the same thing. They're different peptide sequences, but we know where they should be on the retention time scale, the same IRT scale even. And so you can actually use those as your retention time standards as well. We also know the retention time standards for many of the different peptides in the human proteome already. So we know what their appropriate IRTs ought to be. And so you can use just those peptides as your retention time standards are as well. We're actually at the point now where realistically, in very few circumstances do you actually need heavy labeled isotope peptides to be able to use as retention time standards. You oftentimes need to use them as quantitative standards, but in certain circumstances, but that's not really what IRTs are about anyway, right? We're just focused on monitoring retention time in these cases. So here for retention time alignment, you know, we're at the point now where you don't really even need to consider adding spiked in peptides into your samples, unless you, for some reason, need to monitor those types of peptides for another type of reason. So again, these types of peptides you don't even need to necessarily include them. But if you do put them in, I recommend putting them into whichever samples you're interested in monitoring. So put them into the same sample, injected into the same sample, so you have a certain amount of them. Essentially, this can be a nice kind of online quality control if you have these peptides around. Again, they're not necessary, it's just if you have them around. So is there a difference between gas phase fractionation and DIA and just DIA from Susanna? This is a fantastic question. It really is just a limited retention time window. Mass range, as you say. It's really just taking the mass range. Instead of going from 400 to 1,000, you're going from 400 to 500. And the second injection, you're going from 500 to 600, 600 to 700. Everything is exactly the same. That's what makes it great for building libraries, because all your fragmentation patterns are exactly the same. It's the same matrix, so all your retention times are very precise. So this becomes a much more popular, practical way of doing library generation, at least in my mind. And then I've essentially stopped building DDA-based libraries. While they're oftentimes used by a lot of the other types of search engines, this type of DIA-only library generation I find to be very practical for virtually every type of experiment that I do that's DIA-based. So is there any type of software that we need for doing gas phase fractionated DIA? Encyclopedia, the tool that our lab creates, actually has all of the GFP DIA analysis stuff built into it. You can do some of this work in Skyline, and that's something you can ask Lindsay about afterwards. But it is possible to do some of that work in Skyline. But we recommend oftentimes just building, even if you're going to go down that route, building the libraries inside of Encyclopedia and then exporting them for Skyline, because the tooling there is just designed to create these libraries from gas phase fractionated samples. Marco asks a fantastic question, which is, if you're building from hundreds of samples at the same time, is it too diluted at this point? for any given peptide that you're interested in measuring. And if your peptide only exists in one of your samples, then yes, it's probably too diluted. But that peptide is probably not very useful in terms of trying to do quantitative analysis across all of your different runs if it only exists in one biological replicate in one of your samples. It's just not going to be statistically useful going forward. So really, this is actually enriching for peptides that are likely to be statistically useful in your experiment going forward. That said, if you have two very disparate types of experiments, so for example, one experiment I was doing was looking at CSF from two different types of sources. So one is from a lumbar source and one is from ventricular source. These are whether you collect it from the base of the spine or whether you collect it from the back of the head. And there, because of the sort of difference in the type of matrices that those things have, they have very different types of proteins on them, whether it's from the spine or from the back of the head or the spine. So because of that, we actually oftentimes will acquire two different libraries, one for the lumbars and one for the ventriculars. So that's a fantastic question. And we'll analyze the ventriculars as one block and the lumbars as another block. So if you've got two very different types of samples, so for example, if you're trying to compare two different cell lines or something like that, then that's a great way to go.
So unfortunately, we're out of time. But Brian, do you mind staying around a little bit and answer the questions in writing?
Yeah,
You
so
can
absolutely
type the questions while we start already the next part of